My name is Sandrine Hoyt. I'm delighted to welcome today our LCN speaker, Firat Güder, who is a senior lecturer in bioengineering at Imperial College. So Firat has a very eclectic training. So now he's in bioengineering, but he was a research fellow in a group of Professor Whitesides at Harvard in the chemical side of things. Then he did his PhD before that in microsystems engineering at the University of Freiburg in Germany. And prior to that, he trained in computer engineering at the University of New Brunswick in Canada. So quite a broad set of skills there, which is super exciting, and all with hopefully a theme around nanotechnology. Firat's research has been recognized by many prizes, including international scholarships from KU Leuven, I have to mention, also a connection to Belgium here, and the German Research Foundation to fund his trip to the US. Most recently, he's been awarded the University of New Brunswick Young Alumni Achievement Award. And his research is really groundbreaking as much on a fundamental level and also applied. So he's got numerous patents and I think also companies. So we're really excited to have you here today to share your research. I think this talk will be recorded and will be posted on YouTube, on the LCN YouTube channel, and maybe also on the güderresearch.com YouTube channel. Who knows? We're all over YouTube these days. So thank you very much, and we really look forward to hearing your talk on disposable sensors for food, agriculture, and healthcare. Thank you so much.
Thank you very much for the extensive introduction, and delighted to be here today. So I wanted to say that just this morning I realized that this is supposed to be a half-an-hour talk, and initially it was a longer talk for me that I prepared. So I start clipping things down. So I hope that I didn't clip from places where it just creates a lot of discontinuity. But like, do bear with me, and then and ask questions at the end. And if I did, make something unclear by clipping down too much, I should be able to address them. So it's a great honor to be here today, so I'll try to describe some of our research in the area of disposable sensors, and present to you some of our latest work in this area. So to begin with, I want to define the term disposable sensors. So what are they really? So in a nutshell, a disposable sensor is a single-use or a short-term monitoring device that would allow us to make a measurement. And these kind of devices are ideally used or ideally suited for chemical or biological sensing. And this is mainly because of contamination. So we don't want to worry about decontaminating things all the time. Ideally, we want to make them low cost, and then we perform a measurement. And then once they serve their purpose, we dispose them. So, and point of care devices tend to be such as the glucose sensor that's used by diabetics, or the home pregnancy test. These are famous disposable devices. Point of care detection of or monitoring of several various bioanalytes. So and wearable devices, these are mainly the emerging ones, the tattoo-like devices. They tend to be disposable, or something as simple as really pH paper are some examples for disposable sensors. So as I mentioned before, there are so many things I could have discussed and shared with you today, but I had to kind of clip it down. And this was, by the way, the idea was to present these three things in the beginning before clipping down, but let's see. I may not be able to go too deep in detail. But so today I will really try to cover three of our most recent work and hopefully show you a sample of the kinds of things that we really do. So one of the first things that I'd like to share with you is it starts with a bright and colorful image, which is the picture of a supermarket. Although, you know, they at some point last year, they didn't look like this. Shelves were quite empty and so on. But generally speaking, supermarkets, they solve a huge problem for our society. And it took many, many years of development to come to this point where essentially you can go into a supermarket and then buy whatever you like and leave you don't have to chase after an animal anymore or do really much preparation yourself. Everything is pre-packed. It really presents a very picture perfect image that shows, I guess it gives kind of like a wrong signal to us that we kind of solved most, if not all, of our food related problems in the world. But this picture, it again it's a false picture that or a beautiful picture to a pretty bad story, pretty inefficient story. So in fact, to create a system where we can go and buy pretty much whatever we want, we waste a lot of resources. And these resources, for example, even the food that we produce, they never get fully recovered. For example, a third of all food that's produced for consumption gets lost during production and throughout the supply chain itself and the end users. So in fact, 60% of all food that's thrown away by consumers is completely okay to consume, and yet we still throw it away. So one of the reasons why people really throw away food, especially consumers, is the concept of use-by dates, and even this whole thing is quite confusing. There's the use-by date, the best-by date, and then the expiration date. People are using different conventions and so on. Pretty confusing. But what it is, what these dates really are, is that they are a very conservative estimate of how long a food product can be consumed. So they are static, so they do not change with time, and therefore, you know, if you leave, for example, a piece of chicken on your counter, you know, for an extended period of time, the time on the printed use-by date will not really change on the packaging. However, we know that the product will last longer because it's been exposed to the temperatures outside. Obviously, to compensate for these potential risks, the use-by dates tend to be quite conservative. But even then, products are usually safe to consume for much longer period. So ideally, we would like to come up with technologies that would allow us to replace these use-by dates with dynamic tags that would give us some sort of an indication of food quality and you know, whether or not a product is safe to eat. So this is one of the things that we have worked on in the recent years. So and we developed a paper-based electrical gas sensor for monitoring food quality, and the sensors themselves are printed sensors. We can print them in large amounts. They are essentially zero cost. They only consist of cellulose paper and carbon ink. And in our lab experiments, we were able to produce them approximately 2 cents per sensor. But when scaled up, we estimate that the price will go down to probably 10 to the minus 5 cents per sensor. So, and this is quite important because at the end of the day, the packaging industry requires very, very low cost sensors to stay profitable. And this is a very it's a good alternative to the static use-by dates, and at the same time, they're not really going to break the bank in terms of manufacturing for the packaging producers. So let me tell you a little bit about how these sensors actually work, and then I'll tell you later the mechanism and so on. So these are electrical impedance-based sensors. So in essence, again, it's just a piece of paper with carbon ink printed on it. And by measuring so there are no other additives. So, and by measuring the impedance of paper, we can measure the concentration of gases that are in the surrounding environment. And I'm going to explain how these sensors really work, because they appear to be too simple to be true. So, and this idea kind of emerged out of a very simple plot, which I came across in a paper making book, probably from the 30s or 40s, actually, pretty old book. So, and it showed this plot where the moisture content of paper is plotted against relative humidity on the x-axis, and at modest relative humidity of 50%, paper contains approximately 5% water by weight. And I realized that this has significant implications, because although paper appears to be dry and it feels dry all the time, it is in fact quite wet. So there's a lot of water. That means that we can do wet chemistry with paper without ever adding a drop of water to it. So, and really the entire idea behind these class of sensors is that paper is always wet. And we want to really exploit this property of paper. So the way we sense gases is that if we have a water-soluble gas in the environment, this comes and interacts with this layer of water that's really adsorbed on the cellulose fibers. And by measuring the impedance of paper, in essence, we're really measuring, because cellulose itself is not a conductive material. In essence, we're measuring the electrical properties of the layer of water that's adsorbed on the cellulose fibers. So if a water-soluble gas is in the nearby environment, it dissolves in this layer of water and dissociates into ions, and then this changes the electrical impedance of paper, meaning the layer of water adsorbed on paper. And the sensitivity of these sensors are highly correlated to the Henry's log constant of a gas. So if a gas heavily dissolves in this layer of water, then then the sensor is more sensitive. It produces a higher signal. So and we tested the sensitivity of these sensors to ammonia gas, and we were able to go down to as low as 0.2 ppm of ammonia, and as soon as we saw this, we knew that we're onto something quite interesting, because this is almost at the same level as really high expensive sensors, and we were able to already get quite high sensitivity out of these really low cost sensors, and we could potentially go down to lower actually concentrations. However, the precision of our instrument was limited to 0.2 ppm. That we couldn't basically dilute the gases any lower. And the sensors, as I described, they're sensitive to water-soluble gases, ammonia being the most water-soluble gas. So it produces a really high signal, and with an incredible dynamic range, because we are almost limited by the solubility limit of a gas for these kind of sensors. So we tested these sensors for ammonia, trimethylamine, and carbon dioxide. And carbon dioxide, being not so water soluble, produced the lowest response. And then we also tested the sensors with less water soluble gases. Essentially, almost no, they're not water soluble at all. hydrogen sulfide and carbon monoxide, and in the range that we tested them, we were not able to get any signal out of them. So they are not basically these sensors are not too sensitive to these gases. And we use the sensors for the detection of or monitoring of spoilage of fish and chicken. So when protein-rich products they spoil, they release volatile amines and ammonia. So because the relative humidity inside a food package is pretty much fixed at 100%, so the changes in the impedance of the sensor really indicates the presence of these gases in the surrounding environment, so inside the package. So, oops, sorry. So we basically put the sensors inside a package that contains either water or chicken or fish. So and water is really our control, and we were able to go. We were able to monitor the spoilage of fish and chicken using these sensors. So the sensors are sensitive enough to really produce an increasing signal over time as these products degrade. And of course, at the same time, we also ran microbiological cultures to correlate the signal generated by the sensors with microbial count. So, and these are indicated by the bands, I guess the pinkish band for chicken and the purple band for fish. And the sensors were able to essentially produce a signal that would be sensitive enough, or the sensors are, I guess, sensitive enough to be able to detect a microbial or the spoilage levels low enough that would be that would allow basically users to detect levels of microbial contamination. So, of course, we have done much more than this, and I don't have time to cover all of this stuff. But one of the things that's also quite interesting with these sensors is that at the end of the day, these are chemi-resistive sensors. So, and we bought a near-field communication tag. So, and these tags are used in, for example, contactless payment cards, bank cards, Oyster card, etc., or even like our Imperial ID cards uses this technology. So, and we were able to connect. So these cards, they just really have a loop antenna and a chip, silicon chip. So when there's a reader nearby, it powers the chip, and the chip then transmits information back to the reader. But if we put the sensor in parallel with this chip, if there is a gas within the environment, when it reaches a certain threshold, majority of the power delivered goes through the sensor itself, and not the chip. So the chip stops working. So that means the tag stops working. So using a trick like this, we were also able to produce on and off type disposable sensors with these low cost wireless tags. So and the tags themselves cost approximately 2 to 5 cents. So again, I don't have time to show you the videos and so on that we captured, but we have material on our website concerning this technology. Do feel free to check it out. So we started with really the end of the supply chain, and where we're interacting with end users, and maybe potentially even maybe not fully end, but producers can use such a technology as well. But again, there are so many problems with the entire food system that wherever you look, there's something to there's a problem to solve. So there's no shortage of problems. And one of these other problems is the use of nitrogen fertilizers. So the use of nitrogen fertilizers has increased by 600% in the past 50 years. And they are, in fact, the most used soil additive in agriculture today. But the thing about these fertilizers is that their application has been poorly understood. And growers often only follow guidelines. I mean, they don't really make any measurements. So there are various guidelines produced by governments and so on that say, you know, put X amount of nitrogen fertilizer per hectare to if you're growing, let's say, wheat, winter wheat or spring wheat or whatever. So, and because the guidelines are quite static, there's really not much information concerning what's in the soil and how much should be added or not added. So, and because of this inability to measure the concentration of nitrogen in soil very inexpensively, there's a huge variation between farmers, between nations in terms of the use of nitrogen fertilizers. So an interesting stat is that 8 times more nitrogen fertilizer is used in China than, for example, Australia. Just the Chinese government produces different guidelines for their farmers than the Australian government. So it's absolutely not clear at all how much fertilizer should really go into soil. And because of this, excessive use is really the norm. And we just put too much because we want to ensure that there is enough nitrogen in the soil. And lab testing is incredibly uncommon. So, so how much fertilizer do we need to add? We don't really exactly know because we don't really make measurements. So can we determine somehow what are the soil nitrogen levels now and what are the soil levels, nitrogen levels in the future? And can we make then the proper addition of soil nitrogen or sorry, nitrogen fertilizer into the soil to basically stay at the optimum levels to support plant growth? So we have developed a series of technologies for determining soil nitrogen using disposable sensors, but then we combined this approach with machine learning to basically produce or predict the levels of soil nitrogen in the future, but also because the soil nitrogen consists of mainly ammonium and nitrate, we tried to develop a technology that would allow us to measure some of these, but and then predict the others. So really the whole idea behind this approach is that can we measure things that are easy to measure, and then can we predict the rest? And I will describe this, I think, a little bit better by, so my students actually created a video that explains this whole concept. So essentially, to measure soil ammonium, we have invented a low-cost technology, which builds on the food sensor that we had developed previously. So, and how the sensor works is that instead of having just simple paper with no additives, we just add a bit of acid to the paper, and then once the ammonium that's in soil volatilizes, it reduces the conductivity of our sensor, because it essentially neutralizes the acid that's already there. So, and this approach really allows us to but again, nitrogen in soil consists of two main components. One is ammonium, and the other one is nitrogen. There is no really low-cost way of measuring nitrogen. So then we said, well, can we actually use machine learning? So we took really easily accessible data, and such as rainfall, and we combined this with pH, machine learning, which allowed us to measure, predict the levels of nitrate predict the instantaneous levels of nitrate, and now basically, and also this approach allows us to predict the levels of nitrate and ammonium into the future. So, and the instantaneous predictions using the parameters that I have described. So we were able to get a pretty linear correlation between the standard measurements and the prediction or the predicted results with an R-squared of 0.7. And in the machine learning community, really anything above 0.6 is pretty good, especially for a system like this. So which shows that we can actually get pretty good estimates of levels of nitrate in soil without really making a nitrate measurement, and we think that this is a pretty remarkable result because we don't depend on additional hardware to make such predictions. And in fact, if we use in these experiments here, we use the data generated by our sensors to really predict the levels against a standard measurement. So if the inputs are actually the sensor or data inputs are not from our sensors, again, our sensors are low-cost sensors, so their performance is often not on par with standard laboratory measurements. But actually, if we do not use the sensors that we produced and rely on laboratory measurements and then make predictions, then of course then we get an R-squared of 0.86, and the hardware, actually the best hardware out there, produces about 0.9. So we're already getting very close to predicting levels of nitrate without measuring it. We're getting very close to dedicated instruments using this approach. And the results I showed, they were predictions now, also predictions of nitrate levels now, so the instantaneous nitrate levels. But again, using our approach, we can also predict the levels into the future. So essentially, all we do is, of course, I didn't describe this whole scheme and how we develop the models and so on. But all you would really need is a single point measurement of soil nitrate, soil ammonium, and pH and conductivity, and then using this information, predict the levels of nitrate, and then predict the levels into the future. And using our approach, we're able to predict the levels 12 days into the future, which we think actually can improve significantly in the future with more data and longer data sets, etc. And we're able to get an R-squared of 0.64 for levels of ammonium into the future, and 0.7 for levels of nitrate into the future. So I'm getting very close to already my half-an-hour mark. So the last thing I want to briefly go over is our diagnostic device that we have developed for monitoring and detecting infectious diseases. And infectious diseases, they've always been around, but outbreaks are becoming more and more common, especially in the since 2000 we have had already 5 large outbreaks, and partially one of the probably the biggest reasons is the travel. So more people are living in close quarters in cities, in high density areas, and not only that, they travel quite a bit. So and one can travel from one part of the world to another within a day. So, and because of this, really diseases, they can move around really fast. So we have a higher probability of having outbreaks. So and really, this was just so the COVID pandemic. It seemed like a surprise to many, but in fact, it was really coming. So because it just, the right pathogen with the right properties had the potential to really explode very, very quickly, and we're now going through that. So we have developed a disposable silicon-based device, and that would allow us to, I guess, so PCR nowadays, everyone is kind of familiar with this technology. PCR is a method for really detecting the genetic material of a pathogen. So and we have developed a chip that would allow us to perform PCR measurements on the go. So instead of relying on centralized laboratories with expensive instruments. So again, I have a small video that explains this concept, what we have done. Use this chip. So this chip is called Trisilix, and really what it does is it aims to miniaturize conventional tabletop quantitative PCR system into a small chip to eliminate dependence or reduce dependence on centralized laboratory. And this device consists of a sample reservoir that contains the reaction reagents and sample, and a chip, silicon-based integrated chip, which really implements the modes of operation. And on the top, we have an electrochemical take acid used bottom, really electrode silicon. Chemical protection really exploits the interaction of electric molecules and so with these molecules. So as we increase the number of copies of nucleic acids in our sample representation, these redox elements interact with the and they no longer become available. Current use this electrical reactor. The silicon substrate can be used as a heater. It's passive. Again, this is for PCR. We need heating, and that's basically and the thermal resistive properties of silicon as a semiconductor allows us to use the piece of silicon itself as a temperature sensor. This way, we can really maintain our sample at a temperature that's needed to perform the PCR reactions. So, and we chose silicon because it's a highly processable device material, and intrinsic because the semiconductor it can work as a high quality thermistor, so it really becomes an active substrate instead of a passive substrate. And silicon is actually an inexpensive material, contrary to what most people believe. Large wafers can be purchased for as low as $10, and then many devices can be made on a single wafer. The thing is that the only disadvantage of silicon is it's quite expensive to process. And that's really what we tried to solve. So can we, so the question was, can we produce silicon devices low cost, in a low cost fashion in a standard laboratory? So, and we said, well, let's try. We can replace patterning with photolithography with laser cutting. Deposition can be done through mainly wet methods or thermal bonding. Etching, again, would be through wet methods, and dicing to make the chips could also be done with laser cutting. So we basically wanted to avoid all cleanroom-based processes. And when we can produce these chips in a standard lab on a 4-inch silicon wafer, and the chips with all the reagents and everything, they cost approximately 30 cents to produce. So again, everything is produced in a standard wet lab, in my lab, essentially, in the Bessemer building. So we use these chips for qPCR, or I guess micro qPCR, and we compared it against a commercial qPCR device for the detection of a pathogen called Mycobacterium avium subspecies tuberculosis. Don't worry about the pathogen itself too much, but it's a bacterial pathogen, and we're interested in it for various different reasons. But I guess the message here is that our chip works as well as a tabletop device, if not better, and we were super surprised by this. And it costs basically nothing, the fraction of a tabletop device. So we were able to detect, go all the way down to 20 femtograms of DNA, mapped DNA. And the segment we were targeting was approximately 160 base pairs. So, and this was really a big surprise to us, but this system seems to work as well as a commercial device. And so we were in the beginning of the pandemic, and we were just saying, okay, well, can we also try to detect something related to SARS-CoV-2? So we were not able. SARS, obviously, the coronavirus is an RNA virus. So we didn't have the capability to make the transformation from RNA to DNA, but we just said, can we take some complementary DNA that belong to SARS-CoV-2 and also SARS-CoV, so they're quite similar, and try to detect these and then differentiate them. And in fact, we were able to also demonstrate this, but there's still obviously a lot of there are several steps to be done if you want to work with real samples. These were synthetic samples. And PCR requires several heating steps at different temperatures, and our system really, we tried to demonstrate this because if it works with that, it will work with other emerging amplification methods such as RPA or LAMP and so on, which are isothermal amplification methods. So the sample is really maintained at a single temperature. And it obviously, it's a lot simpler than PCR. So as expected, we did also work with that. Okay, I'm sorry, I'm out of time, so I have to wrap it up. So I would like to thank our sponsors. I'd like to thank Hafiza for arranging this, and the LCN management, and for the invite, particularly Sandrine. And obviously this work was done by my team. So I'm very grateful for their hard work. And if you're interested in disposable sensors, we have recently published a, well, I guess no longer recent now, it's almost 2 years, published a very extensive sensor review on disposable sensors in diagnostics, food, and environmental monitoring. So do go check it out if you're interested. Okay, I'm gonna stop now. Thank you very much.
Thank you very much, Firat, for a fantastic talk and great overview of lots of applications of your sensors. Lots of claps there. And yeah, we have time for some questions. I'm sorry that you had to rush, but we'll carry on. Oh, William, you have your hand raised. Is that correct?
Yeah.
Yes.
That was an awesome talk. I loved all of it. I had some questions about the nitrate prediction that you did. When you compared essentially the R value between your sensors and the kind of commercial sensors, it seemed that around 250 parts per million. I wrote it down fast as you were kind of changing your slides.
Sorry, I couldn't hear. You said something. It just disconnected for a second.
Oh, okay. So for the nitrate prediction between your sensors and the commercial sensors, it seems like there was really good agreement for your sensors up until about 250 parts per million, and then it just scatters all over the place.
Yes.
And I was wondering if, I mean, the R squared that you have above that level is really similar to the commercial sensors that you guys tested, it looks like. And so I was wondering if that just means that your sensors essentially don't have enough sensitivity to detect below around 250 parts per million without a lot of variation, or what you thought the cause of that was.
I think so, because we're monitoring with these sensors, with our sensors, we're essentially monitoring a reaction. If the reaction is really fast, right, then I think there is more variation, because we take the way our analytical signal is really is the time it takes to reach a baseline. So I'm speculating here that I think because it takes a lot shorter than when the concentrations are really high, small variations in the concentration when we do the repeats and so on will probably produce a larger spread. But yeah, it's definitely not the same across the entire range. That is true, and we don't really know why yet. We need to probably do much more work.
Okay, thank you.
Yep.
Thank you. Milo?
Hi, Firat. Thanks very much. I enjoyed that. Lots of interesting applications to think about. Maybe one that sprang to mind because I'm forever measuring nitrate concentrations in my fish tank to make sure my fish don't die, is whether I can borrow one of your sensors and use it. Or slightly more seriously, maybe that's another application, including in fish farming for your sensors.
Yeah. So should I answer that one, or you have more questions?
Then I have a technical question as well, but
yes,
you can answer that one if you like.
No, ask the technical question.
So
I can do both.
Yeah, I was wondering I mean, you just said, oh yeah, we print them on paper, but I mean, paper is many different things, and so I'm wondering what how the sensor response, the sensitivity and the selectivity depends on the nature of the paper you select, and to what extent you've explored different substrates.
Yeah. So well, we use pure, essentially cotton cellulose paper, because and we use actually like lab filter paper. And the reason why we use that is because we don't want any additives in paper. For example, if we buy a copy paper, it often has all kinds of other stuff in it. So to modify printability, because you don't want your copy paper to be too hydrophilic, because otherwise, as soon as you put the ink, it goes everywhere. So if you want high resolution, so you want your paper to be a bit more hydrophobic. So and yeah, they would definitely influence the characteristics. And to eliminate these, we just essentially use only cotton cellulose filter paper, so that we don't have any interferences. It would make a difference, definitely.
Mm. But I could imagine that you could have lots of papers which might all be pure, but still have very different textures, because the cellulose could be fibrillated to different extents, you know, all the way down to, 5 nanometer fibrillated cellulose, if that's what you wanted. And so naively, this is the LCN, I'm assuming that the finer you fibrillate the cellulose, the more water you incorporate, and the larger the surface area for interaction with gases. And so I'm guessing you might get a faster response time and possibly more sensitivity, but I don't know whether that's true.
So it's okay. There are a couple of trade-offs. When you have a lot, so one of the things that we know for sure, the density of paper makes a big difference. But so when humidity changes, obviously paper needs to either absorb more paper or absorb more water or desorb more water from its surface. And if you have a lot of water, then it takes longer to actually get rid of this water. So so there are essentially various trade-offs. But your assumption is absolutely right. So the type of paper, like if you use nanocellulose paper, it will have different properties than if you use standard, I guess microcellulose paper. The porosity of paper makes a difference. The density of paper makes a difference. And by density, I mean like essentially grammage per square meter. That's one of the metrics that's used. So and I guess even the treatment of paper would make a difference if there are different sizing agents, etc., that are used for production. We haven't done much work with nanocellulose, but in fact we have recently ordered some samples to play around with. So so I can't really give you an answer on the true effect of using not nanofibrillous cellulose.
Okay, I look forward to the next talk then. Thanks.
Thank you.
Thanks. I think James has got his hand up.
Hello. Yes, thanks very much. Yeah, very nice talk indeed, Firat. Thank you.
Thank you.
I was just a query about, say, ethene, being able to detect ethene, for example, and ripening of fruit and things like that. Is it the problem that ethene is not very soluble in water?
Yeah. So that's one of the issues. I think it would depend on the levels. So we haven't tested so much with for example, like ethylene. Like we keep on hearing about this. And partially the reason is really, or the sensors that we have, they don't really work too well with organics essentially. So we tried various organic solvents. We do see a response, but usually that's upwards of a couple of %. And they also act really and so on. And yeah, I guess the shortest answer is that yeah, it's not very water soluble, and therefore we haven't really explored too much of that yet. But it's something that we keep on hearing.
And can I just add another question? I noticed that you don't really have at the moment very much sort of chemical reactivity involved in your systems of recognition in the recognition event. Is that something that you're looking into?
Yeah, so the first thing really we did was we started like simple, just acid-based reactions. So and they already produce, they provide some level of recognition. And then obviously intrinsically, again, these sensors are sensitive to water soluble gases pretty much only, so that already provide, like that essentially already narrows the range of gases that we can detect. So and other than that, we are also working on a few other approaches to improve our sensitivity, and in fact we're also working on some chemical amplification strategies for gas sensors. So but one of the things about gas sensing is that really the right type of sensor needs to be chosen for the right type of gas, and we think that by keeping it narrow is actually quite good for us then, and try to broaden up for the kinds of applications that we want to do anyways. Because most gas sensors that are out there, they actually don't work really well when there's a lot of water around. And ours does exactly the opposite. It works really well when there's a lot of water around. So we want to kind of exploit more of this. But yeah, I mean, do get in touch, James, if you have some ideas.
I have, and I will.
Cool.
I will indeed. Thanks.
Sounds good. Thank you.
Okay, thank you. Yeah, lots of discussion. Any more questions or any? Yeah, feel free to post in the chat as well, because this stays on. I just had a quick question, because there is when you see disposable, you think about the packaging as well, and maybe sort of tuning the packaging to make it less environmentally damaging, and I was wondering, things like polyethylene and so on, will your sensor enable us to replace them with something less polluting and so on?
Yeah. Well, these sensors themselves, I think they wouldn't necessarily replace them, but people are coming with various packaging materials that would essentially be, so if they're hygroscopic enough, then I can see an alternative where these sensors could directly be printed on the packaging material itself without, introducing additional, like let's say a layer of paper or something like that. So really the idea is to, with this one, is to reduce the waste of food and not necessarily fix the packaging problem completely. Because if we waste less food, that means that we will need less packaging to begin with, but it doesn't directly address the packaging issue, which is a big problem on its own, obviously. So many, again, like so many problems to solve around food. It's just unbelievable.
Okay. Yeah. Okay. Yeah. Thank you. Milo's got his hand up.
Yeah, I can ask another question if there's time. I was interested in the role of temperature. So you showed us, the nice dependence of the relative humidity on the temperature, but presumably also the gas absorption depends on temperature. Mm-hmm. And so presumably you can increase increase the accuracy of your measurement by determining the temperature, which perhaps you can do just by measuring the resistance of your carbon track at the, not between the electrodes, but within the electrode or something. Can you also use it as an integrated temperature monitor?
Yeah, that's a very good point. So I guess if we want to be really accurate, then obviously knowing the temperature would make a difference. So when we measured the sensitivity or response of the sensors to ammonia, for example, at different gases, but it was in a very narrow range between 20 and 30 degrees. Interestingly, we did not really see a huge impact of temperature on the response that the sensor generated. At least they were not statistically significant. But they will certainly. We know that if we go to much lower temperatures, we expect to have a better sensitivity. But this integrated sensor idea is, I think, it's a very good one actually. So it's something we haven't explored too much yet, Maida.
Okay, so I look forward to your next talk again.
Thank you.
So thank you.
Please do reach out. Yeah.
Yeah. Thank you so much, Firat, and to everyone for joining today. Thank you so much.